Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute.
From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on November 27th, 2024. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 69th episode of Matters Microbial. I appreciate every listener and every viewer and every email and or comment. Thank you. At the time this podcast is being recorded, my microbiology students are off for Thanksgiving break before the big push into the end of the semester. One of the most important topics I teach is about antibiotic resistance, since it is a part of most of our lives and the rise of antibiotic-resistant microbes a continual threat. A few years ago, I came up with this exercise to teach my students about this particular topic. If I have a petri dish supplemented with the antibiotic ampicillin and then spread sensitive E. coli over it, those cells do not grow, but they are not dead. If I spot a tiny amount of bacteria that are resistant to ampicillin and are also marked with a fluorescent tag into the center of the plate, over time the students can see satellite colonies. These satellite colonies are not, in fact, resistant to ampicillin. The fluorescent center is the resistant bacterial strain. You see, this type of resistance is due to the secretion of an enzyme that breaks down ampicillin. The satellite colonies do not glow under ultraviolet light because they are not truly resistant. Instead, they're taking advantage of the strain that is destroying the antibiotic all around it. Now, this is clearly relevant to the treatment of disease, and it fascinates my students because of how graphic it is. As we get closer to the end of the semester, some of my Micronauts in Bio 350 make extra credit creative projects. I kind of show them the way with 3D printed glow-in-the-dark earrings, seen here worn by a former student who's now in PA school. Knowing my love of bioluminescence, one student in the past made this diagram of a cephalopod complete with glowing microbes. It warmed my heart. I wonder what my current students will create, and I'm happy to share their work in coming episodes. I have long discussed how microbiology is perceived by the public, devil microbes that are always bad versus angel microbes that are always good. And the truth, honestly, is neither. But there are many fermented foods that are seen as good, and they are indeed the product of microbial metabolism. Which brings us to probiotics and probiotic microbes. There's much discussion of possible health benefits. All one has to see is an advertisement for Activa. In fact, it's so much in the public eye that the Giant Microbes company makes plush versions of bifidobacterium and lactobacillus, seen here. But how to determine the truth about probiotics and their impact on our microbiome and our health? So it's a genuine honor to welcome an expert in this area to the podcast, Dr. Maria Marco of the University of California, Davis. Her research group explores many aspects of this important area, and I'm excited to share her insights with all of you. Welcome to the Quality Quorum today, Maria.
Hi, Mark. It's so great to be with you here.
Well, I mean, I really love the fact we've already talked about this, that your first and last initials are the same as mine. So you and I and Mark Mandel are in a special crew. Let's just be honest about it. I think a good place to start is how you kind of came to this area of research yourself, just to let listeners and viewers know there are many paths to the microbial sciences.
Yeah, yeah, absolutely. And absolutely something I also try to share with my students that there is usually not a straight line from when you are in high school or start college to what you end up doing for the rest of your life. I would have never guessed, honestly. Now that I think back on it, it makes sense. But at the time when I started college at Penn State, I was a biology student, and there were like 1,500 freshmen, first year students, my first semester biology course. I really felt like a number. And Mark, you'd appreciate this is the time when we put our social security numbers down as our student IDs. Imagine that. But so I realized I needed something a little more personalized. And that's when I landed on the microbiology major. And gosh, the rest is history from there. Because I just saw all the potential. I didn't know if I wanted to do environmental science or biomedical science. Um, but I did want to explore research as a student. And so I just like, cold call, knock on doors, email, what you got to do, right? And a lot of rejection. People just don't have time. But then I landed actually in my research experience as an undergraduate in a food science lab. I was a microbiology major. I didn't really think about food science. And actually, the work I did there was on Cynorhizobium meliloti, who this microbe forms, nodules on legumes and fixes nitrogen, right? So kind of a funny situation for a food science lab, but again, it's all kind of universal, right? From doesn't maybe matter so much which department you're in, but it was there that washing the glassware, washing test tubes, 4-hour shifts. I remember, it was manual labor, but it was really fun to be in a space where people were excited about uh just something, right? And just excited to be together, learning together, exploring together, asking creative questions. A postdoc and the lab manager, they were fun, sort of cynical, but having a good time. And yeah, that was really where I said, maybe research is something for me. And again, that was in having to do with the soil and and plant growth. Um, and then after I graduated, I actually worked at the NIH in the Dental Institute for a year, and I studied oral biofilms. That was amazing, kind of solidified my interest in research, but I also had a little more fun time, downtime. And this is a long story, Mark. I hope you're okay with that.
It's not a problem at all. You do know that I did my PhD work with Cynorhizobium meliloti. I was one of
Another thing we have in common.
I was Sharon Long's first graduate student. So
Oh,
yeah.
wow.
Well,
Wow. Wow.
no. I don't know I don't know how I ended up in there. Let me tell you. I mean, everyone I went to grad school with is all famous and everything. I'm delighted to be the microbial evangelist that I am. But as you mentioned, it's a long, strange trip, to borrow from the Grateful Dead, to get
Yeah.
there. Who did you work with at Penn State on Rhizobium?
Karen Miller.
Oh, I know Karen Miller.
She
Sure,
Okay.
that's cool,
Okay.
right?
She's a fabulous scientist.
I
Uh-huh.
tell people that microbiology is a small world, and I'm not just being punny. It's also true.
It is. It is. It puts so many different in ways you don't imagine. So, again, like when you're just take opportunities as they come and go
Yeah.
network and talk with people, right? Because eventually they're still going to be your peers, or you're going to work with them potentially someday, right? So don't close doors,
No.
right?
And you know,
All right.
it's interesting
Hmm.
because you went on to do work with the oral microbiome, which is a
Yeah.
fascinating topic too. And people kind of get like I'm not a popular dinner party participant because of how much I enjoy talking about microbiology. But, you know, the thing is if they give me a bad time, I just lean into the skid. I don't know if you do, but that's kind of my thing. But the
Hmm.
important thing here is you went through a series of different choices of where you were going, and then that ended you up in food science eventually.
Yeah, let me just take you through the rest of that journey there
Sure.
because I think it tells a broader picture here as well. But uh, to talk about my PhD, I worked with Steve Lindau at UC Berkeley, and that's where I studied Pseudomonas syringae, which is a plant pathogen, but also has a whole other lifestyle. It just lives on the surfaces of plants, on plant leaves, and doesn't cause harm.
Mhm.
So I was asked as a grad student to look into how does it adapt to grow and persist on this kind of nutrient desert, and it's exposed to sun and wind and high and low temperatures. How does it manage there? So that's what my PhD was about, and a really fantastic mentor in Steve Lindau. But then still not food science, right? So I actually moved to the Netherlands. I After I graduated, I never would have thought, right? People see me here, Maria Marco, they may think I'm speak Italian or Spanish, and I'm half Italian, but I don't speak either. I speak Dutch. I can talk. So, yeah, for personal reasons, I moved there, married a Dutch guy, and lived there for 6 years. And that brought me then into the fold of the study of lactic acid bacteria, food fermentations, probiotics. There they are, yeah. And again, using network people that we knew and just cold calling, I found my postdoc and then eventual uh, research scientist position in Wageningen in the Netherlands. Fabulous group of people doing amazing research on these different topics. And yeah, so don't look back from there. 2008, when I moved to UC Davis, the food science department made the most sense for me.
Mhm.
It just happened to be by chance that my first research experience was in a food science department, and but yeah, it's been quite a journey, and I think the bigger picture is that I really like to think about microbes as microbes, not as pieces parts. So I think that's, if I look at my research past and what we're doing now, I think that's what's driving me in the way I think about these research questions we have.
You know, Maria, that's a wonderful story about I when I use this term, we can say either pachinko balls through life or drunkard's walk or chemotaxis with tumbles and runs. The point is, very seldom do we make a straight line to something that we ended up at. And I think it's important, as you mentioned, to open yourself up for that. I also wanted to say the sociology of labs is very, very important. I teach at
Mhm.
an undergraduate institution, so I don't have technicians and postdocs and graduate students. I kind of miss that aspect of like the morning confab around the pour over fancy Pete's coffee, right? That's where I learned to drink 14 cups of black coffee a day.
Yeah.
I don't think I have any caffeine receptors in my brain, but you become part of a crew and it's supportive and wonderful. And I have to say that I hear a lot about competitiveness in science, and I'm not saying it's not there. It is. But I have always been made to feel welcome in microbiology. And it sounds like you had much the same kind of approach. And that's wonderful to
Yeah.
hear. Yeah. And
Yeah. We
and
mentor is how we were mentored. Yes.
right. Yeah. And the other thing is, isn't it funny how we don't wash pipettes anymore? I've been talking about this for years. Subject for another time, but that's it. Anyway, it's really good to hear about this aspect of things, and I want to make listeners and viewers know that some forms of food science are about the things that we don't want in our food, and that's very important work on, for example, foodborne pathogens. Arturo Casadevall doesn't like it when I use the term pathogen. That's a story for another time, but you know what I mean. In any event, though, I think it's important to know that the idea of the more positive things. You mentioned, for example, working with Steve Lindau's group and working with things living on the phyllosphere or philosphere of plants. But I don't know if listeners and viewers know that when you make your own sauerkraut or kimchi, you're counting on microbes that live on the surface of the plant to carry out the next stages of the fermentation that creates that fermented food product. So it's all connected.
It is.
And it's all ecology too. So one of the things that I wanted everyone to think about, and you're the ideal person to talk about before we go to microbial brass tacks, is the idea of the way that probiotics is talked about in the media. And it's very easy for me to get judgy about it. And I want to listen to what your point of view is, because you had mentioned that you have this, I'm not going to call it a love affair. You have a crush on lactobacillus. and I feel the same way about some of my favorite microbes. So how do you feel about the whole probiotics question?
Yeah. Well, great question. Yeah, I guess I want to take it from maybe a historical perspective, and then from onto that to what can we say at this point to the future. So just about the time that we were learning about all of the pathogens that cause infectious disease, and that was, starting with the great work of Pasteur and Robert Koch, and then the contemporaries, just in a, amazing time. Uh, Really critical for public health. There were also scientists, microbiologists, immunologists pointing out, hey, not all microbes are going to be bad. And I think of Eli Metchnikoff, who got a Nobel Prize, by the way, in immunology, was pointing out that hey, there's microbes in yogurt, and by chance, it seems like the people in the Caucasus Mountains may live longer, and maybe these microbes we have in yogurt can kill off the putrefactive, nasty microbes in our intestine, and we can, live to be 140 or something like that, right? That's didn't say that, but putting that on for color. So interestingly, even some of the first human studies where they're having randomization, kind of the development of the practice of human clinical trials in the early 1920s, were performed on Lactobacillus acidophilus. What? And at that time, the findings from the studies, and these were the early studies, but still, like, there were benefits from consumption of these microbes. And then we move on a bit, and we have the isolation of E. coli Nissl, which is sold as a probiotic, and not in the U.S., but in some other countries. To in Japan, developing this fermented milk by this company we may know as Yakult, with lactobacillus, and that's like in the 1930s, right? So this has a long history, a long trajectory, and kind of along the road, where this type of research or interest has really grown has been maybe a little bit more on the commercial side, kind of seeing the potential for the application of microbes, just as we would in making a cheese or a yogurt, preparing the microbes for consumption. So it kind of blended hand in hand in that. But then we reach the 2000s, and where we're having this explosion of information about the microbes that inhabit our bodies, our gut microbiomes, our skin microbiomes, and so on. And the pretty amazing connections we're finding between the microorganisms that colonize our digestive tract and our immunity and our metabolism and our moods, like in pretty convincing and um, amazing and fascinating ways that kind of captured the consumers, clinicians, and so on. Um, the field of probiotics research has been developing along that 120 year trajectory, with some pretty good studies, and I'd say there's even evidence. We look in kind of a putting together different clinical studies in a meta-analysis and systematic reviews. And for certain indications, probiotics are efficacious, right? There's a moderate to at least benefit of taking a probiotic. Um, so the challenges we have are not maybe not surprising, because there's a lot of interest in improving health in many ways, treating disease or preventing um, future illness. And so, there's like 250 some registered clinical trials on probiotics in every kind of category. And then you layer on top of that, we're talking about different strains and different strain combinations, and even different delivery formats. So it's a very complex question, and that is meaning that we may not have all the answers, that there will probably be new and better strains or strain combinations for probiotics in the future. Um, but the more I think about this, and the more you mentioned fermented foods, right? There's some of the same microbes we have in a sauerkraut, or a yogurt. And so I'm starting to see a bigger picture emerging where, yes, we're going to have the importance of our resident microbes and feeding them the right things and having them do the right things for us. But there is a kind of need potentially for us to have just exposures to non-harmful commensal microbes that we've evolved with.
Mm.
And I'd say lactobacilli are definitely in that category.
Oh, I'm very impressed with your answer here, and I'm going to explore it in a couple of different ways with you, if that's okay.
Sure.
I'm automatically suspicious when a company tries to sell a substance with an idea, for example, that a particular type of organism can improve mood. What's interesting to note is there are clinical studies that are suggestive of this. That is not the same as a company selling something, however. Um, so that's one thing.
Mhm.
And I hope we're in agreement on that.
Yeah, and I'm happy to help answer that question or address that concern.
Sure.
I agree. But there's also a regulatory environment that we have that is very constraining. Like, there could be some of the best human studies in the world, um, for a probiotic or for a substance, and they're not able to talk about it as a food when we're talking about a food, because you're not supposed to have health claims on foods. You can have structure function claims and say something flowery like, improves gut health, right? Which, what's that mean, right? So just uh, where companies get into trouble is when they start to make health claims like prevention or mitigation of disease.
Yes.
And so, I hear you, and I am, can be equally skeptical, but it's very hard to combine the two when you have good science with that commercial element without moving into it being a drug.
And I appreciate that very, very much. I used to work in a biotech company where we would use exopolysaccharides from
Mhm.
bacteria as food additives, and the way that that was treated was entirely different than anything I was used to. So that I
Mhm.
think that's really important. I mean, I also go with the idea, first do no harm. When we're eating sauerkraut, for example, or if we're drinking a little bottle of Yakult, which I do, um, you're not really going to hurt anything when you do that. That's important to know. And there's a book, I don't know if you've read Sandor Katz's book, Wild Fermentation.
Yeah, I'm familiar with it.
Right.
Mhm.
And so that the genesis of that book was the idea that it might help people with particular health problems. But one of the points that he makes in the book is it's also good for general health. And I think that we in industrial society probably don't eat a diet that we've evolved to normally eat. So this is why I think fermented foods and the organisms that are part of it, the ecology, for lack of a better term, because that's what you're eating. You're eating an ecology, right? A series of, I would argue, you're eating a series of ecological setups when you eat, like, say, sauerkraut, which is funny to think about. So I really appreciate what you're saying there, and I'm glad that you brought up the regulatory issues.
I do want to come back to this fermented foods connection.
Please.
Um, so just to point out that the word probiotic um, actually has a pretty specific definition, and well, very specific has a definition since 2001 with the WHO, and then some kind of minor textual changes. But the essence of a probiotic is that it's a live microbe that, when applied in sufficient amounts, confers a health benefit. Yeah. And that means we need to know about the strain. We need to know about the genome of this microbe. We need to be able to track it. And when we make a sauerkraut, there aren't probiotics in sauerkraut. You may see that on your label, on the label of the sauerkraut container. That just, I think, signifies that there could be, no promise of, but could be live microbes in that sauerkraut. Just as you have live and active cultures in yogurt.
This is really important to say. And as a scientist, I guess I can call myself that. I really appreciate it because there's a difference between, and I'm going to go ahead and say this, the ecology and the members of that ecology. And if we're talking about health benefits, we darn well better understand the constituents of that community and what roles each of them play. In a way, and this is an unpleasant metaphor, but when you think about fecal microbial transplants in the treatment of C. diff, it's important to know what organisms are conferring the benefit, not just the source material that's being transplanted, for lack of a better term. So this is very
Mhm.
similar in a way, and I really appreciate that distinction. Thank you. Maria, is there a reason that when you take a probiotic supplement, you have to continually take it as opposed to just once?
Yeah, great question. So these microbes are not generally going to colonize. They might persist for day or week, but they're not going to replace the microbes that are already living in our digestive tract. Um, Particularly with people that are um, otherwise healthy and don't have severe like disease, where the gut microbiota would be depleted. So yeah, you wouldn't expect that the probiotics are going to engraft or permanently
Mm.
colonize. And so I kind of think of that as a good thing, because I may not want a microbe to just colonize forever. There's could be unintended consequences. Um, and that's why, if you would, you for probiotics, you would want to have them regularly.
Related question, Maria. If they don't colonize, is there competition taking place, or is it just a matter of who can acquire turf within the gut?
Yeah, absolutely. Of course, there's competition. These are, I don't know, microbial uh, communities that have already been established. They got their thing, and it's very hard to introduce a microbe into that established
Mhm.
city, established community. So what we found actually is that diet can have a factor in how long a microbe or lactobacillus can persist in the digestive tract. So interestingly, however, that's not really been explored in human studies. So diet has been kind of absent from probiotic clinical trials. But maybe that should be included as a factor because, as we know, the diet we eat definitely impacts the microbial metabolism and community structure in our digestive tract. And so, why not a microbe that we ingest? So that may provide some opportunities to kind of see this nuance in persistence times or colonization potential of a probiotic. That we are, Lactobacilli and Bifidobacterium, or that are two main groups of microbes used as probiotics today. But does that answer your question?
It does. I mean, I'm aware of how, and forgive the triviality, if you view us as a tube
Yeah,
and we take in things at one end and things are eliminated at the other, we have things that are permanent residents. We
correct.
have things that are just passing through, for lack of a better term. And then we
Hmm.
have, and I'm going to call them squatters, and these are things that set up shop there temporarily, or
Hmm.
sometimes with certain types of disease causing organisms permanently. So that business of how they set up shop becomes important. And I know from things that I teach and things that I've read, there are very specific mechanisms that are often used to make this happen. And I don't know that we know enough about that when it comes to the lactic acid bacteria.
There's a couple points here. One is, do we need to have colonization to have an effect? And then, secondly, what exactly are they doing, whether they're colonizing or not? My research, actually, when I was a postdoc, showed that lactobacillus is metabolically active in the digestive tract, at least the
Amazing. Yeah.
species that we commonly have as probiotics. So they can survive through the stomach into the small intestine, and that's this important site in our bodies because there's a lot of immune sampling.
Mm.
There's a shorter period of time that they're there, but they are going to be metabolically active. They reach the colon. And so I see it as that maybe don't need them to colonize, but that they do have the peptidoglycan, they have the lipoteichoic acids, they are metabolically active, they are breaking down even lactose in if you have them in a dairy. So
Mhm.
uh, I think it's a little bit of a simplistic view to think that they have to colonize to have an effect.
Very good. No, I really appreciate that comment. Thank you. Maria, some people who are really into the idea of taking probiotic supplements, there's an argument that you should have a diversity of different organisms that you are consuming versus one or two. Do you have thoughts on this?
Mm-hmm. I understand that the idea is like throw everything at it, the kitchen sink approach. Um, I think the jury's out a bit on whether that really helps, as opposed to just having one strain. We know when you get groups of microbes together that they interact and can affect each other. So, what happens when we start to put multiple strains together? We don't really know. I think it's kind of wishful thinking in a way that that's all like gonna they're all gonna work synergistically. Um, so I think that's still the jury's out as whether it's gonna be doing more, or you know, compared to if you have more together or if you just have one strain.
I mean, I hope you don't mind this image, but sometimes I like to think of the gut microbiome or microbiota, more accurately, as this incredibly diverse forest. And so that's a lot to overcome and a lot to understand. And sometimes we only see, if you'll forgive this, the end product. Um, you knew where I was going with
Absolutely.
that, but
I
yeah,
work a lot with poop.
there you go. Yeah. Um, One of the things we've talked about quite a bit on this podcast is the idea of communities working together. And I think you may have already addressed this to some degree, but the idea is, how could just a few additional components interact with that community? What you had said before is the idea they don't have to persist to have an effect. I particularly appreciated your term about immune sampling taking place as the gut does.
Yeah, yeah. I kind of liken it to, we estimate you only need to have consumed 10 cells of enterohemorrhagic E. coli to get sick.
Yeah.
So why then are we so challenged to think that having a billion lactobacilli will have no consequence?
Now, this is really, really well stated. Thank you for that. It's how I discuss virulence in class, and I
Mhm.
know I'm not a medical microbiologist, but the idea it takes just a couple of cells, say for Bacillus anthracis or botulinum, to have this horrific effect, versus Salmonella, where you have to consume a substantial amount of those to have the
Mhm.
disease effect. Um,
Mhm.
so there are some organisms that have a huge effect from a very small number, and others that have less of an effect, where there needs to be more of a community. That's the way I would see it. It sounds like
Yeah,
we're onto
absolutely.
something.
Mhm. Mhm.
So finally, and this is a kind of a blue sky kind of comment. What Not everything that we find is easily grown or easily scaled up for commercialization. What on earth could we do if there was an organism that was found that would be a fabulous probiotic, but we couldn't develop it as something that could be used as a supplement? Do you have any thoughts on that?
Mm. Yeah. Well, where there's a will, there's a way, usually. So certainly there are uh, now companies dedicated to producing strict anaerobes in large scale, more indigenous members of our gut microbiomes for use as um, next generation probiotics or live biotherapeutic products, another name for a probiotic, really. Um, so there are challenges to that, but you already see some products come out with, like, for example, acromantia uh, being one. Um, so I guess I have less concern about that being available. I think the bigger questions are going to be, again, of course, regulatory, and then also strain variation. You've done such a nice job on your program here explaining what happens when you grow microbes in the lab. And then we have also the strain vary between the genotypes, so different strains. And that's just to bring it back to standard old lactobacilli or bifidobacterium probiotics. That's been a big issue in the field, is the strain variation. I think we're going to have the same issue when we look at Akkermansia and Faecalibacterium and Clostridium and so on for health benefits. Strain will matter. So I see, the technology we can reach. It's the bigger microbiological questions that we'll have to be wrestling with.
It's so interesting to me because I tried to come up with like an epigram about this, but the hand of Darwin is on all that lives. And we tend to not really understand that we are exerting selection at all times, no matter what we choose to do. And we tend to forget that. And these organisms are not in business to do what we want. They're in business for themselves. Now, I know that they don't have their own perception, although I've been accused of secretly believing that, which is true. But you know what I'm getting at. And I think that this is a real issue when you look at the complexity, how to tease these things apart. And I don't even think the people that are all knowledgeable about systems biology would claim that they could tease apart all the different parts of the intestinal microbiome.
Fermented foods bring in, I think, some other really important attributes that if you buy a supplement, you're not going to get. right? So I'm not saying it's either or,
Hmm.
um, but I am saying that we are starting to put together a picture that when you make a fermented cabbage, I think as Ben Wolf was pointing out, that there's going to be some variations, going to be dependent which microbes are on that cabbage you start with, or your ingredients, and maybe even your kitchen.
Yeah.
um, and so there's going to be this variation. But what is also true, and Ben also pointed out, is that pretty frequently you get a fermented food that you can eat. Right? There might be little changes between them, but it's remarkably consistent. We're not adding in microbes intentionally as we would if we're making yogurt or a starter culture for cheese or so on.
Hmm. Hmm.
What we've been looking at more recently is there sort of a metabolite composition or composition to the fermented food that can be healthy. So it's not just the microbes, but it's the products of their metabolism, the breakdown of the nutrients, the fibers, for example, in cabbage, or you could pick another food, right? Or the making of new things like vitamins, and other compounds like lactic acid, acetic acid, that get into this fermented food. Is it this whole package that when we eat it is really going to benefit our health? Um, and what we're finding is that if we take a commercial sauerkraut and we make a sauerkraut in the lab, we know they have different microbes, same species, right? Generally, right? Lactic acid bacteria, we know their lactic acid ferments, but they do come together with sort of a very similar profile that's very different from the cabbage, the raw cabbage that we started with. And when we test this package of microbial products on epithelial cells, intestinal epithelial cells, we see that fermented cabbage, sauerkraut, is protective of the gut barrier. So it can help reinforce barrier function, at least in the lab, is what we're seeing. Whereas the raw cabbage, same way we prepare, there's really no benefit. So looking at the metabolites that we see in a fermented cabbage, compared to the raw cabbage, there's a lot of really cool things in there, phenolics, but also some compounds that were being discovered through mechanistic research on gut microbiomes, to be beneficial. So maybe just a little bit of sauerkraut is going to go a long way, is where I'm going with all this.
That's fascinating, though. And one of the things when I'm sad I don't get to teach as I'm slowly fading away into retirement. I don't get to teach intro bio, but one of the concepts I love teaching students about are emergent properties, where you bring together two things you wouldn't
Mhm,
expect have a
mhm.
result that you like. For example, deadly chlorine gas and explosive sodium metal together make table salt. And in a similar way, I suspect that that's the situation in the complexity of our gut, is that you have lots of organisms that are working together in ways you couldn't have predicted from their individual natures. I know that there are some types of, and I know I can't say Bacteroides anymore, and I haven't learned the new name. I'm still stinging from Sinorhizobium changes. Okay. It's a problem for me. But I do know that there are some of these microbes that live off sugar acid in mucus secretions in our guts, and they're actually inducing the production of more mucus. And I say they're kind of like mucus farmers, and we're the field. And it's an interesting way to look at it, because we're a series of ecological niches that are used by different organisms in different ways. I am particularly interested in the idea of things that co-evolve together versus things that are incidentally together, because we swim every day when we're walking around. It's a sea of microbes in which we normally live, right? So this
Mhm,
is a
mhm.
fascinating story.
Mhm.
And the barrier results that you talk about especially interest me because of the relationships to things like irritable bowel syndrome and similar sorts of disorders.
Yeah, yeah. These are sort of biomarkers for how these metabolites are going to be received by our intestinal epithelium, and that is so many consequences along immune system, gut brain axis, metabolism. Um, so we are, I think, kind of emerging into this age of diet, age of personalized diet, personalized medicine, of course. And so I think what another kind of project I've been working on is like, what is gut health and how do we define it? And a lot of this will be impacted by what kind of diets, what kind of exposures we have. Um, if I could talk about one other thing here, with foods and microbes, another project I've been involved with, it's through the International Scientific Association for Probiotics and Prebiotics. It's a mouthful, but it's ISAP for short. It's a nonprofit, science leaning, focused organization on biotics. And for a few years, we've been exploring the question is what if you just ate more live microbes? Or just ate more microbes. It's kind of, you said, this exposures of the sea we're exposed to. And a lot of our immune sampling, so looking for microbes and antigens, comes in from our intestine, our the mucosal associated immune system. Um, and it's been kind of interesting. We've like kind of brought in this idea, probiotics, very specific strains, and then we recognize that fermented foods like yogurt and kimchi are going to have thousandfold, so more microbes than lettuce, right? And is there just these exposures that we should be having that uh, can benefit our immune system early on and throughout our lives, and similarly our metabolism? So we took a look in this database called NHANES, National Health and Nutrition Examination Survey. Okay, I
Cool.
think that's pretty much it. um, Very important nationwide survey looking at diet and metrics of health. About 5,000 people are surveyed at a time, and they're collecting dietary records. And so I had the pleasure, along with several others, to look through 9,000 food codes in NHANES database, like people reported eating, like pizza or frozen peas and meatloaf, right? And cataloging how many microbes are going to be on those foods when they're
Oh.
eaten. Yeah, that's a challenge, right? So we didn't say, oh, there's, 1,000 cells per gram, but we went kind of low, like
Mhm.
pasteurized milk, and so 10,000 microbes or less per mil or per gram. Medium, which would be like a apple or
Mhm.
salad, which have microbes, maybe in the 10,000 to million cells per gram range. And then the fermented foods, which would be, a lot more. And well, it wasn't so hard to look through those 9,000 food codes because most of them would be in the low. 96% of the foods have very few live microbes. And so, we think about that, and the foods we've been eating over the past, 50, 60, going on 80, 100 years, they're clean, they're safer, but they also have fewer microbes. So
Mhm.
maybe just introducing more of these fermented foods or probiotics into our daily lives will give us those exposures that we want or we need, as opposed to we want to avoid. And that may help us. We have correlations suggesting that eating live microbe foods is helpful, from looking at the NHANES health metrics. but we need to do actual clinical trials, human studies to tease this apart.
It's very important for, and I run into this all the time with family members, when they hear about study X and can't
Mm.
they apply that to their own lives, and it's difficult because we're such individuals. We have to have a large study size. We have to do it in a controlled fashion to know whether or not a particular treatment or a particular condition can have a particular response. And that doesn't come naturally to a lot of people. I'm not being negative. It's just most people have other things to do with their time than think about those things. Uh, I know so many examples where there appears to be evidence of something wonderful from a particular microbe, and that researcher gets pelted with questions about obtaining that microbe. And the researcher has to say, we don't know what the effects are in a large group. And I think that that's so important to do. But that's why you have this large group. You're trying to approach that question.
Right. Absolutely.
I'm enjoying this session so much. And I want to explain why, because you've said several things now that made me think about my own preconceptions. The
Mm.
fact that I'm using the term probiotic inaccurately. And I really appreciate that correction. When people use the term a probiotic, you're talking about specific organisms where we know a little bit about them, as opposed to fermented food, which is a galaxy of
Mhm.
different microbes. And as you mentioned, and this is 100% true, you can make two different sauerkrauts side by side and have different subpopulations. So I really appreciate that further information, which I will use. I think that's really good. Um, Lactobacillus, you adore that organism. Tell everyone why they should adore that organism.
I think that we were just talking about fermented foods. So lactobacilli are needed to make these wonderful, diverse fermented foods. We coexist with them, and uh, I think that there are the group of microbes that we have today for which we have the most information on how they interact with us as people or as animals. But you're never going to answer all the questions. There's so many. So, just for example, um, in 2020, there was a revision of the taxonomy of the Lactobacilliaceae. And there were at the time over 260 different species of lactobacillus, and that number has grown since then. But they were so diverse, they didn't belong in the same genus. So now we have 24 different genera of lactobacilli, and they were given, unfortunately, or fortunately for us, have to talk about it, some complicated names. So Lactobacillus plantarum is now Lactobacillus plantarum. Yeah, so it's a mouthful. We still have Lactobacillus as a genus, the type strain, Lactobacillus delbrueckii subspecies Bulgaricus, for example, the one in yogurt, Lactobacillus acidophilus. So there's still the genus Lactobacillus, but now we have 23 other genera. So that's how diverse they are.
Mm.
And kind of in matching with that, they're found um, in our digestive tracts, they're found in our mouths, they're found in vaginal tracts. It's like 99%
Hmm.
of the microbes in the female vagina is like lactobacilli.
Hmm.
So there they are. They're in animal digestive tracts. They can even reach higher numbers. They're not very abundant in our intestines, but they are in pigs, for example. They're found on plants. They're really important in insect digestive tracts. So we live where we're surrounded by these, I hope that doesn't gross you out, but these kind of friendly microbes
Mhm.
that used to just be called lactobacillus, but now have some new genus names.
It's interesting. I've been fascinated by the way that media talks about microbes, and there's a lot of negativity, but sometimes there's some positive stuff. But when you look at many of these microbes, they evolved with us. And I mean, they're part of
Mhm,
us in essence,
mhm,
we don't do so well without them. Incidentally, I mean, that's the funny part about it. And but people get so carried away because of the. And I actually have this to show you. It's this whole business of germs.
mhm,
And when Pasteur used the term germ, what he meant was a piece of life. He was using it to defeat the
mhm,
idea of spontaneous generation. He just meant a piece
mhm,
of life. And this life is everywhere. And it doesn't mean that when you have these living things that are on you or in you, that they're negative necessarily, as we've been talking about. Some of them have extremely positive effects. I used to routinely have students isolate lactic acid bacteria from between their teeth, that kind of thing. And they're
mhm.
and we got them just like we get them from cheese, right?
Yeah.
And they're part of our lives. And I think it's interesting. And I won't even start talking about kefir with you, because that's a subject I could go on for a long time about. I have many questions about kefir, but that's another story. But this is a good reason to use as kind of almost like a mascot for reminding people about the positive aspects that microbes can have. Um, where I used to teach, there was the cafeteria. There was a source of yogurt, and we were the ones in my class to discover it was not a monoculture of different lactic acid microbes in there. Clearly, it had been like people had brought stuff from home, and it had like evolved over time. So I wanted to name those the whole yogurt collection after the institution, but they wouldn't go for that. So but this is really good to know. And you've also, I think, had a wonderful time during our podcast discussing some projects that your own group has been involved with. And I thought, and I bring this up, and I'm not trying to waylay you in any way. I get accused of that sometimes, but not by you. Let me see if I can pull it up. When I was reading about your work, I saw this wonderful thing that came out in July of 2024. Health benefits of kimchi, sauerkraut, and other fermented foods of the genus Brassica, which is the only way that I will consume Brassica, by the way, as part of a fermented food, but that's me. And this is a wonderful paper, and I'm going to give a link to it in the show notes. Did you want to say a couple of things about that particular work?
Yeah, sure. I really have to give the credit here to the first author and the team that wrote the paper, Sabina Fijan. She contacted me a couple of years ago. She's a professor in Slovenia, and
Wow.
yeah, we hope to collaborate. And we she really led this initiative. And what I really like about the paper is that it puts together the different human studies that have been performed on this group of vegetables. Um, and that's really important because otherwise we don't have kind of an overview of where the science stands.
Mhm.
Um, Yogurt would be the most studied fermented food in human studies, epidemiological, so observational and randomized controlled trials. And then I would put kimchi second to that, and mostly because of studies that are performed in South Korea.
Right.
Um, so we've started to look at kimchi with collaborators in South Korea, and it's like sauerkraut, full of some really exciting microbial metabolites that could put together this picture of how eating fermented brassica, like sauerkraut or kimchi, uh, can benefit metabolism and reduce risk for weight gain and type 2 diabetes. So I think we're going to start to see some convergence in this research in the next 10, 15 years or so as, we start to look at the molecular microbial interaction side all the way through to what happens when we eat these foods.
I sometimes call microbes the small masters. Okay. Alite domine pabule, all hail the small masters. And there's some truth to this. Isn't it interesting that this kind of fermented food that's been around for literally thousands of years, we're now finding out have all these possible benefits that we hadn't really known about, even though folks used to talk about it without being able to study them closely. And
Yeah.
this business of how nature teaches us things is a very interesting one to me. And it must be a lot of fun to work on.
Oh, yeah. Yeah. Never thought I'd be making sauerkraut in the lab, though, honestly. That was not how I started. But
No, no.
don't,
I'm
yeah.
my wife, Dr. Jennifer Quinn, chides me every day. I don't care for leftovers. I have real issues about this stuff. I'm one of those super tasters you've heard about. Um, I've made yogurt. I've made kefir. That's about my limit of making my own fermented foods. So there you go. But
Yeah.
I'll eat sauerkraut. Yeah.
Yeah. But going back to your point, like we have been eating these foods for thousands of years. And one of the first questions you asked is, how did I get into this, a business? And I still get a lot of inspiration out of my grandmother. She would make pickles
Yeah.
every year. And she wasn't making them because they were going to be healthier. I think she liked the flavor, right? And they're a way to preserve the cucumbers that she was growing in her garden. So a lot of the inspiration, I think a lot of the connection in microbiology is talking about foods and talking about um, bringing in fermented foods in particular, because there's so many traditions around the world of making fermented foods from pretty much every kind of ingredient. And that's so translatable because then, you can notice the difference between the substrate, the ingredients, and the product. And so that kind of serves as that gateway into talking about the benefits of microbes and how we coexist with them. We have this project called Eat Lac, and with that project, we invited anyone who's making fermented fruits and vegetables to send us a small sample of their ferment, and we reported back on which microbes are in there as a a way to acknowledge like these home fermenters, people who are
Mm.
making these foods in their kitchens are really scientists, right? Just the process of cutting up, chopping, salting, packing, all of those things, changes in temperatures have huge impacts on the community of microbes that grow. And so I think that's really been a valuable piece to ending up in a food science department, teaching undergraduate, graduate students, and sharing this with, my grandparents, for example, and my neighbors, and people who are just generally making this connection between health and diet and microbes.
I think it's so important to note, and we've talked about this, how different our diet is the last 100 years or so. And I know that prior to 100 years ago, there were a lot of negative effects. I know that. And there were a lot of bad things too. I mean, I tend to
Hmm.
see the world more gray than most people do, I suppose. But
Hmm.
it is really true that we've changed the way in which we consume food and deal with it. And the microbes are part of that. So coming back to the whole idea, can eating microbes improve our health? I would argue I don't see how it could hurt.
Right.
And of course, when I don't mean I don't mean like take a bunch of things that cause disease. That's not what I'm saying. But the idea that it has been a part of our diet, and it's something that people have a negative a negative viewpoint of because of bad PR. I don't know if you know this, but I often send my students out to look for what I call swab stories. I'm making a joke about sob stories where they'll say, I swabbed this surface and there were microbes on it. And I'm going, well, yes, but it's put in this negative way. And it really isn't that. They're part of our universe. They're part of our they're part of our biosphere. They're part of our bodies, as you've mentioned before. And so it's important to keep that in mind. I wonder if you have some advice for our listeners and viewers as we start to wind things down. And I only say that because we've talked about a lot of things that people are going to be thinking about as they open their refrigerator doors.
Yeah, I would like to comment on what you were saying though about
Sure.
this exposure, to microbes and kind of tongue in cheek, but really like when we started this project thinking about live microbes in our diets, you're kind of approaching it from maybe there should be a recommendation or an adequate daily intake for microbes. And maybe, that is a missing piece to our diets. So, but with that, right, and also the point that they're not going to be harmful. I always, explain that if somebody's very is immunocompromised, undergoing treatment, or in the hospital, to avoid these types of foods. Um, but what we're doing now is, in a way, kind of just trying out different ways of getting microbial exposures. You may decide to try a probiotic supplement. Not promoting that. I'm just saying that it may or may not work for you. And that's going back to the differences in our physiology and our microbiomes. The same with foods. We shouldn't eat a food if we hate the taste. We're not going to stick with it. So the food has to taste good.
Yeah.
And so there are so many different kinds of fermented foods. And also, Mark, just to point out, to acknowledge the different traditions and cultures. I've had an opportunity to work with Abiai Hauptmann, who's Greenlandic Inuit, and others, exploring this question of food sovereignty. And the way fermented foods and other foods, a part of indigenous cultures, have been viewed as, in very negative ways, as being even unsafe. And so I think this also opens up opportunities for conversations about how we respect other people's foods ways, and kind of learn from it, really, acknowledge and appreciate and learn. So that's kind of one headspace I'm in. And then the other, which we didn't maybe get to today, but just thinking down to the very detailed level of the metabolism of lactobacilli, and their fermentation metabolism. So another area of interest, and the kind of from the big picture down to understanding the very mechanics of lactobacilli and how they do what they do.
I think the good part about what you just well, first off, I want to say this idea of ethnomicrobiology is very interesting to me. And I know perfectly well what you're talking about the idea of, I'm going to say, casting aspersions on other cultures. And I don't mean to turn this into that kind of discussion. It does exist. And I do think we need to respect, other cultures' approach to cultures. If you don't mind my saying so, it's really true, because we have something to learn from that. This isn't something that people came up with in the last 15 minutes. Some of those cultures have thousands and thousands of years of experience of using that particular microbially associated product. So there are things to learn from that, and it may be lost over time. And so that's why it's important to pay attention to it. So I really want to showcase that comment of yours, which is excellent. The other the lactobacillus, and I'm not going to learn the new name quite yet, is certainly very genetically tractable. And that means there are all kinds of approaches from my end of the bench, as it were, to make changes and learn about how things take place. And that we can start to tease apart how these things work and the interactions between them. And that's really an exciting thing to think about. So I have learned a lot today, Maria, and I want to thank you personally. I mean, I always get quite excited about these sessions, but I knew today was going to be particularly exciting because at the time I'm recording this, Thanksgiving is tomorrow, but it's certainly an important part of things. I also was really touched by you bringing out issues of family, because certainly my grandmother used to do a lot of pickling and I miss her a lot. So this is interesting. I could have had conversations with her about this if she were still with us. But I really do want to wish you and your research group and friends and family the very best. And if we're going to be thankful for anything right now, we should be thankful for the microbes around us.
Absolutely. Thank you, Mark, for all that you do. This is really, really an important program. And so clearly you're an outstanding educator. I wish I had you as my instructor. Wonderful.
Well, thank you so much for your kind words. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Maria Marco is professor of food science and technology at the University of California, Davis. Many, many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile each time I hear it. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.